Hello learners, myself Vineet Kala. I'm Assistant Professor in Department of Microbiology, KPP College, Vashi. I'm a microbiologist by profession. I've cleared CSNet and today I will be talking about prokaryotic gene regulation and we will be focusing on Lac Operon. So the contents here are for this lecture is we will be covering definitions, regulatory regions of operons, we will be covering negative regulation and only overview of positive regulation. By the end of this session, you will be able to describe the steps involved in prokaryotic gene regulation, explain the roles of activators, inducers, and repressors in gene regulation. We will be also discussing about a mechanism of negative and positive regulation, so you will be getting a clear idea. Now, definition. So, first thing, what differentiates prokaryotes from eukaryotes is its DNA. Okay, in prokaryote, the DNA is in nuclear form or it is said to be in naked. So, and it is CCC, supercoiled DNA. What do you mean by CCC? So, CCC means covalently closed circle, and it obviously DNA is supercoiled, highly supercoiled. If you measure the length of the DNA, it is around, for example, Escherichia coli. It is measured in centimeters. But when you compress or when DNA is compressed, it is in microns, less than a micron. Okay. Now, what are operons? Operons are functioning unit of DNA. It consists of clusters of genes, which consist of regulatory regions. Uh, yeah, and there are structural genes. So basically, operon consists of regulatory regions and structural regions. So if I compare, if I can give you an analogy of that, everybody has a pipes in home and it will supply you water, right? Now, how can you control the flow of water? It is by using taps, right? Now, if there is no tap, for example, if there was no taps, so there would be a whole lot of wastage of water and water would continuously flow. And the mess it creates, it will burn your energy to clean up that mess. Same, likewise, organism has the genes which are under control of regulatory regions. Now, these genes should not no fire, as in it should not express 24/7, 365 days, right? Yeah. So, for example, these are inducible operons. Inducible means it is necessary when it is necessary it will be expressed. When it is not necessary it will be repressed. Okay. And there are other genes which are expressed 24/7, 365 days, which are called as constitutive genes. Okay. So there are inducible genes. There is constitutive genes. Okay. Now operon, functioning of operon is to repress the functioning of genes which are not required at particular time. Okay, very simple. Now, example given here is for anabolic pathways. So for productions, so for production of tryptophan amino acid, there is a trypoperon which is involved in production of tryptophan. Okay, there is also operon for catabolic pathways. For example, lac operon, and lac operon is for lactose utilization. Now, in lac operon, the structural genes which are produced is for lactose utilization, that is beta galactosidase enzyme. Now, function of beta galactosidase is to convert lactose into glucose and galactose. Very simple. Yes. Now we will be moving forward to repressor. What is repressor? Now these are proteins which suppress the transcription of a gene in response to external stimuli. For example, if heavy amount of glucose is present in our environment, yeah, so there is no role of lac operon to be functional. Yeah, if only glucose is present in the environment, Lac Operon doesn't have Lac genes, beta galactosylation should not be produced. Therefore, it is closed off. And how it is closed off, we will be seeing through a simulation experiment in future slides and animation. Okay, now, yeah, so there is a repressor. These repressor in Lac Operon, for example, there is a Lac. Now, this repressor represses the transcription of these structural genes. So, you will get a very clear idea by watching animation in future. For just for understanding, I am explaining this part. Now, we have looked upon repressor. Now we will be going for activators. Now, repressor represses the transcription. What do you mean by activators? Yes, you guessed it right. Activators increases the transcription of the gene. Okay, in response to external stimuli. Yes, stimulus. So here, so in Lac Operon, CAP is a general protein. CAP full form of CAP is catabolite activated proteins, and these form complex with cyclic AMP. I will discuss about this cyclic AMP part in future. Now there are third part is inducer. Now inducer, so these are small effective molecules that either activate or represses transcription depending on needs of cell and availability of substrate. Now in Lac Operon, example given is for only for Lac Operon. Okay, so allolactose is a natural inducer for Lac Operon. It induces or uh, so repression is a part of negative regulation. Okay, basically allolactose removes the repression part. How it removes the repressor protein? It binds to repressor protein. So allolactose is a effector molecule, right? And it will bind to repressor protein in the allosteric site. Now, so allolactose, when it binds to allosteric site, it produces a conformation gene in repressor molecule. Therefore, repressor cannot bind to DNA, and it will allow RNA polymerase to function normally. Now, there is also a chemically made man-made man-made inducer, which is called as IPTG. Now, what is IPTG? It is nothing but isopropyl thiogalactose. Okay, and it is non-metabolizable in nature, but it is very much a structural analog allolactose. It looks structurally similar to allolactose. Therefore, it binds to repressor protein and it produces a conformational gene. But main difference part of allolactose and IPTG is its metabolizable nature. IPTG is non-metabolizable. Mind it. So IPTG is basically used in various molecular biological methods for when Lac Z is involved. So you will get a clear idea when we will be watching animation and how Lac Operon essentially works. Now promoter, as name suggests, it promotes the transcription event. Now here the promoter are basically the sites where RNA polymerase bind and initiate transcription event. Uh, yeah, so here example given is lac I promoter, and it doesn't have any other regulatory regions. So lac promoter, there is no operator site, and obviously RNA polymerase will bind and transcribe the lac I gene. There is also next we will be going for operator. What is operator? Operators are basically a main regulatory region, and it binds to so it is a sequence where repressor binds, and repressor represses the transcription event. Here example given is lac operator, and so main part is structural genes also. Structural genes regulate. We have covered regulatory regions wherein promoter and operator is present. Structural genes are basically which will actually produce genetic product, which is protein. So there are three genes in Lac Operon: Lac Z, Lac Y, and Lac A. The function of each gene will be uh, explained in future slide. Now, what is monosystronic mRNA and what is polycystronic mRNA? Simple. From first word, we can guess. Mono means one, poly means many. Cistron, as in these mRNA can mRNA which can translate into one protein is called as monosystronic mRNA. So I will repeat it again. If one mRNA gives rise to one protein, it is called as monosystronic mRNA. If one mRNA gives rise to or translates into more than one protein molecule, it is called as polycystronic mRNA. Okay, I will give example of this in future slides. So this is a physical structure of Lac Operon. Now these regions is present. This Lac Operon is present in E. coli. Okay, and E. coli is obviously a prokaryote, so there is a single chromosome. So these genes are present in single chromosome, and this part and this part of chromosome, this part of gene are present far apart. But for better understanding, we have put it next to each other. Now these are far apart. Genes are far apart. Okay. Now this region it codes for repressor. Lac I gene codes for repressor protein, and there is a promoter for it. Okay, so example, RNA polymerase binds to promoter site and it transcribes the Lac I gene into mRNA. Okay, this is DNA, okay, obviously, and after translation there is a formation of protein, which is Lac repressor protein. Okay, now this Lac repressor protein has this Lac I is constitutive in nature, and Lac repressor is proteins are formed every now and then. Okay, now this this Lac repressor protein has a habit of scanning all the DNA sequence for its target operator site, and it will bind to specific operator. So for Lac Operon, it will bind to Lac operator only. Okay. Once it scans and binds to operator site, it will stick to it transiently, and it will be removed. It gets removed. Not removed, it will fall off. Okay, and it is replaced by another repressor. Okay, so this system continues, and obviously repression is maintained. So how is repression maintained? For example, RNA polymerase comes into the picture. Okay, and this is, for example, operator is filled by Lac repressor protein. Now RNA polymerase has to transcribe this Lac Z, Lac Y, and Lac A gene. Okay, obviously operator site is filled with a Lac repressor, so it is a type of roadblock, and this roadblock is so massive that RNA polymerase cannot move forward. Okay, and if RNA polymerase cannot move forward, it will not transcribe Z, Y, and A gene. Okay, so repression is maintained. So this is called as negative regulation, wherein repressor binding to operator site will form a repression or negative regulation, because these genes are not transcribed, and therefore Lac Operon is closed. Okay, so how can you say so how it can function, how repression can be unrepressed? Yeah, so for we have covered 2 points: inducers. Okay, so herein inducer comes into the picture when environment is full of lactose, yeah, lactose. Now lactose is a disaccharides. It forms an isomer which is called as allolactose. Now allolactose it is an enhancer or effector molecule. These effector molecules can bind to repressor. Now so basically repressor has a DNA binding domain. Repressor itself is a form of molecule of HTH helix turn helix model. Okay, and it has a allosteric site. So allolactose will bind to allosteric site, and it will form, or it will convert, or it will tell lac repressor to go undergo conformation change. So lac repressor molecule undergoes a conformation change. For example, this is a flat molecule and it is binding to operator site. After allolactose binds it to it, so there is a conformation change. For example, like this. Okay, for better understanding, I'm drawing like this. Okay, and this is a allolactose. Allolactose is binding to lac repressor molecule. Now this conformation, in this conformation, this repressor molecule cannot bind to operator site. Okay. Now, if it cannot bind to operator site, what will happen is, now there is no lac repressor. If there is no lac repressor, RNA polymerase can easily bind to promoter site. Yeah, I have to tell you one thing. Promoter and operator sites are overlapping. So if lac repressor is present, RNA polymerase has a difficult time to bind to it. Okay. Now operator is free from lac repressor. Now RNA polymerase comes into the picture and it will easily transcribe this mRNA, lac Z, lac Y, and lac AG. It will form only one mRNA, which will constitute lac Z, which will constitute information of lac Z, lac Y, and lac A. All three genes. Now lac Z forms beta galactosidase enzyme, which will convert lactose into glucose and galactose. Lac Y is lactose permease. Okay, and lactose permease is an protein which will bind to cell E. coli cell membrane. Okay, it will bind to membrane, and it will enhance the transport of lactose from the environment to inside the molecule. Okay, so it enhances only selectively transports lactose from outer environment inside the cytoplasm. Now there is a third a protein also produced, which is transacetylase. It is an enzyme, and its function is still very much unclear. But as the name suggests, it transfers acetyl. Okay, this much is a pretty much Lac Operon, and we have covered negative regulation. Okay, so negative regulation consists of formation of lac repressor, binding of to operator site, and then its removal by allolactose. Now there is a animation, very beautiful animation by Rice University, and it is an open source, and I will be showing this. Okay. Okay, and I will be playing it, and please listen to it carefully.
Upstream from the Lac Operon is a regulatory gene, LAC-I, which encodes a repressor protein. The repressor tetramer is slowly but continuously produced by the cell. Now let's look at the control sequences, the promoter and operator, for the structural genes. The operator overlaps the promoter. If a repressor molecule is present, it can bind the lac operator. It doesn't stick to the DNA like glue, but continually binds and releases, so the operator is...
Okay, so here lac repressor binds to operator, and it gets replaced by another lac repressor. So in between replacement time, RNA polymerase gets a very slight chance to transcribe these structural genes, and therefore when this RNA polymerase transcribes this mRNA, it is called as basal level of transcription. It is explained.
Is not bound by the repressor at all times, and structural genes are transcribed at low or basal levels. When there is little lactose present, there is no reason to waste energy making the enzymes to process it.
Right. So cell requires two things more: energy and obviously resources. So it tries to control most of the resources, and therefore operation operons comes into the picture. So what is the function of operon? It tries to minimize the wastage of resources and energy. Okay.
But what happens when there is a lot of lactose to process and the cell must use it as its carbon source? The
Okay. So
repressor
if for example, if only lactose is present in the environment, now cell has its genes for metabolization, and yeah, only few and therefore E. coli has E. coli at molecular level. It has to enhance the or sorry, lactose breakdown. So it requires all the genes for lactose breakdown. Herein, uh, negative regulation is now removed. Negative regulation is removed. How? By allolactose.
proteins have sites that bind lactose. Lactose binding causes a change in the shape of the repressor so that it can no longer bind the operator.
Conformation.
Without competition for the promoter, RNA polymerase can repeatedly transcribe the structural genes.
So when negative regulation is removed, positive regulations kicks in. Okay, and it kicks in when there is only lactose present in the environment. Okay, lactose is only carbohydrate source. Here, cap protein, as mentioned before, catabolite activator protein, it is present in the cytoplasm. But as only carbohydrate present in the environment is lactose, therefore inner concentration of ATP of the cell reduces, as lactose is not metabolized yet, and there is no other source of carbohydrates or energy. So ATP level drops drastically, and obviously in its the concentration of cyclic AMP, which is a which is inversely proportional to concentration of ATP, it drastically rises. Now, cyclic AMP has a forms a complex with catabolite activator protein, cap protein, and it will bind to promoter cell. Now, what is the function of cap CMP complex? It enhances RNA polymerase binding capacity to promoter region, and therefore, therefore, as the binding of RNA polymerase increases, affinity increases, more number of mRNA is transferred. Okay, for example, if there is only a negative regulation is removed and there is no positive regulation kicked in, like for example, only 10%, only 10 molecules of mRNA is produced. If positive regulation kicks in when cap protein and cap CMP complex binds to promoter site, the transcription level increases 10 to 100 folds. Okay, so this is positive regulation in very much overview.
And translation of the now abundant mRNAs makes the enzyme for processing lactose. The Lac Operon is an inducible system, and lactose is its inducer.
Yes, you can easily view this video. It is obviously present in YouTube, and you can go through this link and you will get this YouTube. Okay, it is an open resource and it is freely available to everyone for access. Okay, these are my references, and I have used majorly animation from Rice University, and I have very much of very good knowledge of thanks to iGenetics, biochemistry by Constant, and biochemistry by nature. And Wikipedia was a source of definitions. Thank you for being there.